Hello and welcome. Today we are going to learn about a very important human disease we know as blood cancer or leukemia. I will be very specifically be talking about two types of blood cancers like ALL, acute lymphocytic leukemia, and CML, chronic myelogenous leukemia. But before I talk about these two types of blood cancers, I would like to give you some background about leukemia in general by answering some basic questions about leukemia. For example, what is leukemia? Now, like any other cancer, here too, a cell undergoes behavioral change with respect to its division abilities and differentiation. And since we know that blood cells are produced in bone marrow, so it is the bone marrow cell which undergoes this kind of transformation or change. What is also seen that leukemia cells are generally white blood cells which are still developing, so they remain in the state of incomplete differentiation and they divide continuously without any control. For example, if you look at a normal blood composition in this panel, you see the blood plasma has different kinds of blood cells like neutrophils, RBCs, monocytes, and platelets. Whereas if you look at the blood of a leukemia patient, here the blood is dominated by leukemia cells, So, and that is because of a cell undergoing a change or transformation where it does not fully gets differentiated into white blood cell. It remains in the incomplete developing phase. Okay. Now, there are different ways of classifying leukemia or blood cancers. For example, depending upon the speed with which the disease progresses itself. In a patient, it can be acute leukemia or chronic leukemia. Like in this case, the disease progresses very fast. And in chronic leukemia, it grows very slowly. Okay. And a patient can have these symptoms unnoticed for years, So this is one way of grouping different leukemia into either acute or chronic. Another way of classifying leukemia is the kind of cell undergoing change. For example, if a myeloid cell undergoes transformation, it leads to myeloid leukemia. Or if a lymphocytic cell undergoes transformation, it leads to lymphocytic leukemia. Now, as you may know that myeloid cells are responsible for giving rise to RBCs, WBCs, and platelets. Okay, so therefore, if this cell undergoes a cancerous transformation, then instead of making RBCs, WBCs, or platelets, it is going to make the poorly developed WBC progenitors. And then lymphocytic cell, they give rise or develop into WBCs. And WBCs are the backbone for our immune system. So, depending upon which type of cell is undergoing transformation, the cancers or leukemia can be myeloid or lymphocytic. Okay. Now, most recent and acceptable classification of different kinds of leukemia is by putting the previous two together. So, in today's time, we classify all the cancers into these four groups. That is, either it can be ALL, that is acute lymphocytic leukemia, or acute myelogenous leukemia, AML, or it can be chronic lymphocytic leukemia, CLL, or chronic myelogenous leukemia, CML. So this is the current way of classifying all leukemia or blood cancers today. Now, this disease can happen from childhood to adults. So there is no specific age of humans which suffer with these disease. You know. Now, the next question is how we diagnose the leukemia. Now, as we discussed in case of tumorous cancers, they can be diagnosed by way of X-rays and CT scan. Whereas leukemia cannot be diagnosed using these kind of technologies. For leukemia, one has to go through the blood checks or complete blood check. Even microscopically, one can detect this disease or by using flow cytometry. Okay, so what I'm trying to say here is that the ways of diagnosis between tumorous cancer and the blood cancers are different. So normal technologies which we use for tumorous cancers are not employed to diagnose blood cancers. Okay, next question is what causes leukemia? Like we saw in other type of cancers, here too, a mutation in proto-oncogenes or tumor suppressor genes are responsible for leukemia as well. So basically, a gene which is controlling the normal cell division, if that gets mutated, it can cause leukemia, or a normal tumor suppressor gene, which is required for keeping a healthy cell into non-dividing state, if that undergoes mutation, that also causes leukemia. So leukemia can be caused by way of mutations into proto-oncogenes or tumor suppressor genes. So with this kind of background, let us look at how the two types of cancer I was referring to, like acute lymphocytic leukemia or chronic myelogenous leukemia, are caused. Now, there is a family of genes we call ABL family of genes, which is responsible for controlling different functions in the related to growth and development in human body, actually. So one of the members of this gene family, like ABL1, this is the gene which makes a protein, which is a tyrosine kinase, actually, scientifically. It codes for a protein, tyrosine kinase, and which is responsible for phosphorylation of target proteins. Target proteins which are involved in cell proliferation and another process we call apoptosis. And both these processes are very important for normal cell growth and development. That is division and apoptosis. Okay. So in ALL or CML, the involvement of this gene is seen. And what is this gene? Very specifically, it is a tumor suppressor gene. It is found that it is a tumor suppressor gene, and I have explained the role of tumor suppressor genes in my previous two videos. But here too, a tumor suppressor gene, its expression is required to keep the cells in a non-dividing state. So keep this in mind. Okay. Now, what happens to this gene? And what is that change which leads to the development of these two types of blood cancers? So this gene also undergoes a mutation. Now, what kind of mutation it undergoes? That is chromosomal translocation. Chromosomal translocation is the key for bringing about a cancerous change in this gene. Now, what happens in translocation? In translocation, one part of a chromosome is transferred to another chromosome. So it's a movement of part of a chromosome from its original place to the new place. Like, for example, if you see in this, if these are the two chromosomes, you know, uh, so this part is exchanged with this part of the other chromosome. And as a result, you see that the one part of B chromosome moves to A and vice versa. And when these segments move from one chromosome to another, they also take along with them the genes which are located in these regions. So these genes, they change or they move to new locations. And this movement to new location actually is responsible for the changes in the expression pattern of the genes. For example, if you see here, we have chromosome 22 and chromosome 9. Okay, so chromosome 22 has a gene we call BCR gene. Okay, BCR gene, okay, which codes for a 160 kD BCR protein. And you have chromosome 9, where we have CABL gene, or what we call, which codes for 140 kD protein, ABL protein, the protein which I was referring, which is responsible for cell proliferation and apoptosis in normal cell, you know. But then what happens that, okay, this BCR protein region, okay, it has two breakpoints. Either it can break here or it can break here. But if you see here in chromosome 9, this ABL gene, it has only one breakpoint. It can break here. So what happens in translocation? When the translocation takes place between chromosome 22 and chromosome 9 among these regions, you know, it leads to uh, the generation of these two kinds of situations. Okay, and this was first seen in patients, or in a patient in Philadelphia. So therefore, this arrangement is also known as Philadelphia chromosome. You know, so what is happening if the BCR gene breaks here and joins with this point? It generates BCR ABL fusion gene, or this kind of arrangement is seen in ALL patients. Similarly, if it breaks here and joins here, it generates this fusion arrangement, which happens in CML patients. Okay, that is, chronic myelogenic leukemia patients will have this kind of fusion protein or fusion gene. Okay, so these fusion genes, for example, in ALL patients, they make a 185 kDa fusion protein. out of which 45 kDa is coming from here, from the BCR part, and the remaining 140 kDa is coming from the ABL protein gene from here. Similarly, in the other possible uh, translocation, you get 210 kD fusion protein, which is out of which 70 kDa comes from BCR region because it is taking a lot more region from here, 70 kD from here, and remaining 140 kDa. This 140 kDa is common, okay? So only thing is the BCR region, if it breaks here and joins, it forms a BCR-ABL fusion gene, which is seen in ALL patients. And if it breaks here and joins here, it forms BCR-ABL fusion, which is seen in CML patients, which suffer with this kind of. So a simple translocation between two chromosomes, that is between 22 and chromosome 9, and these fusion products or fused genes, the kind of protein they make, are responsible for causing this disease. Like if this kind of arrangement happens, or if you have the BCR-ABL fusion, it leads to ALL, and if you have fusion between BCR-ABL fusion gene, like breaking here and joining here, it leads to CML. Okay. Now, what actually happens in both the cases? What you get is a fused protein formed, BCR-ABL fusion gene. The size of protein may be different. In ALL, it is 185 kDa protein, and in CML, it is 210 kDa protein. Okay. Now, but these two fused protein, actually, they are the one which are responsible for promoting cell proliferation, and they also suppress apoptosis. And these are the main cause of cancer development. That this ABL gene in the new location, it undergoes a functional change. It undergoes a functional change that is normally its expression is required, which phosphorylates target proteins, which regulate the cell division and apoptosis. But what happens in its new location when it makes a fusion protein, either in ALL patients or CML patients, it undergoes a functional change. And that change is such that it promotes cell proliferation and it suppresses apoptosis. Means it doesn't allow the changed cells to die, and they continue to survive, which gives them immortality, So that is the you can say molecular basis of these two kinds of cancer. The good news here is that both these kinds of cancers or leukemia are curable, mostly. Like, for example, ALL can be cured by long-term chemotherapies, and CML has more targeted therapy drugs available, wherein we use tyrosine kinase inhibitors. A lot of drugs have been designed, and one classical drug is Gleevec. This is the commercial name, but imatinib, or nilotinib, or dasatinib, and bosutinib. So these are all basically different tyrosine kinase inhibitors, and these drugs are very, very potent inhibitors of this, which takes care of this abnormality which is developed in the cancer cells or the leukemia cells. So to conclude, what I have tried to discuss with you today uh, the the very important disease of humans like blood cancer or leukemia, a general aspect of it, and very specifically talking about two blood cancers like ALL or CML, what happens in these cancers and how these cancers are managed or treated. So I thank you very much for patient listening to this video, and I hope you enjoyed as much as I enjoyed making it for you.